Hello guys, and today is our second lecture of the renal tubular processing, and now we will continue the our previous lecture. So let's get into it. First of all, the PCT and reabsorption mechanisms. As we have discussed that a lot of substances like glucose, amino acids, sodium chloride are reabsorbed in the proximal convoluted tubules. To be more specific, glucose and amino acids are reabsorbed in the earlier part of the PCT, and the chloride is reabsorbed in the late part of the proximal convoluted tubule. And all of these reabsorptions are occurring by the secondary active transport. Let's discuss about this. Let's revise our previous discussion that the sodium potassium ATPases, they pump the sodium out and they influx the potassium into the cell using the ATP. It's the primary active transport mechanism. So this result in the lower sodium inside the cell, and also causes the cell to be more electronegative, about minus 70 millivolt. The sodium in the lumen of the nephron is high, and the sodium concentration inside the cell is low. So sodium will follow the electrical and the concentration gradient and it is moving into the cell by the facilitated diffusion. But it does not move alone. It can carry some other substances like glucose, amino acid, chloride along with itself, right? So glucose will move against the concentration gradient, amino acid will move against the concentration gradient, as well as the chloride will move against the concentration gradient. So it's the active transport, they are moving uphill. But the ATP requiring here is not coming directly, right? The ATP is utilized here in the sodium potassium pump, and it is utilized here indirectly. So this kind of transport is known as the secondary active transport. Sodium is moving by the facilitated diffusion, but the glucose, amino acid, and the chloride is moved by the secondary active transport. So chloride is also reabsorbed by other mechanisms. First is the sodium chloride co-transporter, right, which is following the principle of secondary active transport. Second is the solvent drag. So let's discuss about the solvent drag. A lot of solvents, a lot of solutes are present in the interstitium, right? For example, potassium, sodium, ultimately they will move out of the cell into the interstitial space, right? So in the interstitium, it causes the increase in the osmolarity. Solutes are more and the solvent is less, so osmolarity has been increased. Water will follow the solutes. Water from the lumen will move either paracellularly or transcellularly into the interstitium. It is known as the solvent drag. The solvent will follow the solute concentration. But the water is not alone. A small quantity of sodium may be present in the water, a small quantity of chloride can be present, small amount of glucose, amino acid can be present in the water. So the solutes along with the water will move from low concentration to the high osmolarity. It's known as the solvent drag. And the chloride also moves from the diffusion process from high concentration to low concentration. The second process is the sodium chloride co-transporter, and the third is the solvent drag, in which the water is moving from the lumen into the interstitium, either through the cells known as the transcellularly or in between the cells known as the paracellular routes of the water movement. Talking about the protons and bicarbonates, the normal concentration of protons in the human blood is 40 nanomole per liter. It's not mole per liter, it's nanomole per liter. It is maintained very low in concentration because our body has a specific pH which is 7.35 to 7.45. We will take the average 7.4. Proton concentration, proton is basically H positive, right? So proton concentration is extremely important in our blood, so it is maintained within 40 nanomole per liter range. Okay, And when the proton concentration changes, our body mechanism starts to change accordingly. For example, in acidosis, a lot of protons are there, right? In case of acidosis, proton concentration inside the blood increases and the proton starts to bind with the plasma proteins like albumins, globulins, fibrinogen, etc. And in case of alkalosis, when the protons concentration decreases, okay, they will start to detach unduly from the plasma proteins. Plasma proteins have a specific configuration, either they are held together by primary configuration, secondary, tertiary, or quaternary, right? They are held together by specific bondings, but if these bondings are broken down due to the proton attachment or detachment, it results in the misfolding of those plasma proteins. And if the plasma proteins are not functioning, our body system shut down and it's very dangerous, right? So we need to maintain the pH of the blood within a narrow range, which is 7.35 to 7.45, and it's possible because of the presence of bicarbonates. It's the hero of the story, right? HCO3 negative is alkali. It's the bicarbonate. It is very important to maintain the or stabilize the pH of our blood. But from where that bicarbonate is being produced? It is produced in the kidney cells, in the tubular cells of the kidneys. But how they are going to produce? We'll discuss them in a minute. Our body is a acid producing machine. It means that in all of the processes like glycolysis, in anaerobic glycolysis, lactic acid is produced. Also, in the Krebs cycle, carbon dioxide is produced. Carbon dioxide, lactic acid, sulfuric acid are all acids, but we need to maintain the pH within a narrow range. So, kidneys should make bicarbonates more, right? Kidneys should replenish the bicarbonates if once they are destroyed. Bicarbonate replenishment is a very important process when it comes to the renal physiology because kidneys are the organs that are involved in the bicarbonate production. So let's discuss how it's made. First of all, in the tubular cells, water is available, right? In the renal tubular cells, water is available and carbon dioxide is also available because these cells are also performing the process of the Krebs cycle, right? So carbon dioxide is also available. Water and carbon dioxide will combine to form H2CO3 with the help of enzyme known as the carbonic anhydrase. It's very important. And once the H2CO3 is formed, bicarbonate will dissociate from that and proton will be formed. H2CO3 will dissociate into proton and bicarbonate, right? And this proton, because the concentration of the proton inside the cell is more and in the lumen it's less, so proton will move from the cell outside by the process of diffusion, right? And a lot of bicarbonate is being filtered in the glomeruli. A lot of blood is coming through the afferent arteriole and eventually dividing to glomerular capillaries, and filtration process is going on. This bicarbonate, the filtered bicarbonate, will combine with the proton that has been dissociated from H2CO3 inside the cell. So they will combine and again they will form H2CO3. And H2CO3 in the presence of enzyme carbonic anhydrase divide into water and carbon dioxide, and the water eventually goes down the drain and the carbon dioxide, as the concentration of carbon dioxide in the lumen increases, it will eventually diffuse into the cell, and this carbon dioxide can be help in the formation of more bicarbonate inside the cell. So what we understand from here, we are understanding that this bicarbonate, the filtered bicarbonate, is not the same as the produced bicarbonate. They are different, right? Because the filtered bicarbonate has been combined with the proton and they have formed H2CO3, which have been dissociated into water and carbon dioxide. But we have a concept that the production of the new bicarbonate depends on the filtered load of bicarbonate. When more bicarbonate is filtered, more H2CO3 is there. When more H2CO3 is there, more carbon dioxide and more water is there. So carbon dioxide will combine with more water and they form H2CO3 again inside the cell, and this H2CO3 will dissociate again to proton and the bicarbonate, and this cycle continues. Production of new bicarb in the renal tubular cells depends on the filtered load of the bicarbs. Okay, let's discuss about another type of active transport. Active transport are of three types. First is the primary active transport. The second is the secondary active transport, and third is the pinocytosis, right? Pinocytosis is also a type of active transport because it is also requiring ATP directly. A lot of uh, proteins, we have discussed that plasma proteins do not filter, right? Because they have a charge barrier and size barrier. Albumin is of the smallest plasma protein, but due to its negative charge, it is not filtered. But there are other proteins which have low molecular weight. For example, hemoglobin and insulin are low molecular weight proteins. They are freely filtered, and these are captured by the proximal tubular cells. In case of insulin, insulin is broken down into amino acid, and this amino acid can be taken up by the peritubular capillaries. But in case of hemoglobin, hemoglobin break down into iron, and this free iron, the free form of iron, is very dangerous to the tubular cells, and it results in the tubular cell necrosis, acute tubular cell necrosis. So let's discuss the mechanism of how the pinocytosis is going to occur inside the nephron. The low molecular weight proteins are here. They are attached to the luminal surface of the PCT cells, right? So they are invaginated inward in the form of the vesicle, and this vesicle is formed due to the help of proteins known as clathrins. Clathrins are proteins, and they are occurring their function. They are providing their function with the help of ATP. That's why this process is active transport. Eventually, this membrane starts to invaginate inwards, and it will pinch off from the actual membrane and form a vesicle, right? It's the vesicle. So here we will have the vesicle, it fuses with the lysosome, so the proteins will be break down and all of this process is going to occur. But most important thing here to remember that iron in free form is very dangerous to the cells. That's why iron is always combined with ferritin molecule. They are metal carrier proteins, ferritin. But in case of hemolysis, right, when RBCs get ruptured in any condition, for example, there is incompatible blood transfusion. So the donor RBCs will be ruptured inside the recipient bloodstream. So these RBCs will release hemoglobin, and this hemoglobin, when it comes to filtration process, it will reabsorb by the proximal tubular cells, and it will broken down into iron. Hemoglobin contain iron, right? This iron is very dangerous to the cells, and it will cause acute tubular necrosis, which is a typical complication of incompatible blood transfusion. When there is incompatible blood transfusion occurring, so there will be hemolysis of the RBCs. So these RBCs will release hemoglobin. Hemoglobin is not free in the blood, right? Hemoglobin is present inside the RBCs, and inside the hemoglobin present the iron. Let's discuss another concept about transport maximum. What is transport maximum? When all of the glucose is filtered and all of that is reabsorbed in the PCT. For example, all of the glucose has been reabsorbed. But if the filtered glucose exceeds the maximum transport capacity of the system, the extra glucose will appear in the urine. If this process is transport maximum. In case of diabetes mellitus, when the patient is suffering from hyperglycemia, there is a lot of glucose inside the blood, right? So when the excess amount of glucose undergoes the filtration process, the sodium glucose co-transporter is working. The sodium glucose co-transporter has its maximum capacity, that how much glucose it can handle, how much glucose it can transport. But when the filtered glucose exceeds the maximum transport capacity of the system. System means the sodium glucose co-transport. So the extra glucose, how it gonna move into the cell? It will not move into the PCT cells either. It will move down the drain. It will be appearing in the urine. The patient will have glucosuria. For example, in case of amino acids, when the amino acids are exceeding the maximum capacity of the transport system, the excess of the or the extra amino acid will appear in the urine. For example, homocysteine. Homocysteine is an amino acid, but when the homocysteine level increases in the blood, the extra of the homocysteine will appear in the urine, and this condition is known as homocystinuria. All those substances that are transported or that are reabsorbed from the luminal surface via special transporters, they exhibit the phenomenon of the transport maximum or Tmax. But exception is there. Sodium transport or sodium reabsorption does not display the phenomena of the transport maximum because sodium is reabsorbing via different mechanisms, and its reabsorption is dependent on concentration gradient and electrical gradient and also the renal tubular flow. We have discussed when inside the cell the concentration decreases and inside the lumen the concentration is increased, so sodium will move along the concentration gradient. Also the cell is electronegative, the sodium is a cation, so sodium will move down the concentration gradient. The third thing on which the sodium reabsorption depend is the renal tubular flow. It means when the GFR is high. Wait a second, I will make you a diagram. That this is the nephron, right? And filtration is occurring in the glomeruli, right? Glomerular capillaries. So when the GFR is high, when a lot of GFR is being made, when the concentration of GFR is high, the renal tubular flow will be high. So the speed of the renal tubular flow will also be high. It means that the contact time between the transporter, it can be either of the transporter, it can be sodium glucose co-transporter, it can be sodium chloride co-transporter. So the contact time between the transporter and the solute, we are considering sodium here, right? So the contact time between the sodium and the transporter is decreased, and it will result in the decrease in the sodium reabsorption and the vice versa. If the GFR is decreased, so the renal tubular flow will decrease, and the contact time between the transporter and the solute, in our example it's sodium, it is increased, right? Because the filtrate or the uh, tubular flow speed is decreased. So the contact time between the transporter and the filtrate or the solute is increased, so it results in the increase in the sodium reabsorption. One more thing here to worth mention, that under the influence of hormone, for example aldosterone, in the distal convoluted tubule, aldosterone has a major function is in the sodium reabsorption. But this aldosterone, how it will reabsorb sodium more? It will cause the cells to increase the synthesis of the sodium potassium ATPases, right? Coming back to this diagram, that when the concentration of the sodium potassium ATPases has been increased in the principal cells of the distal convoluted tubules, a lot of them are present, right? So they will function more, they will pump sodium more out, concentration of the sodium inside the cell will decrease more, and it will cause increase in the concentration gradient, and a lot of sodium from the lumen will follow the concentration gradient. So eventually this hormone aldosterone is increasing the synthesis of sodium potassium ATPases, and it will eventually increase the concentration gradient. So this was for today. I hope you understand the concepts related to the renal physiology, and in the next lecture we'll continue this discussion. I will see you in the next lecture.